Welcome again students, welcome to another tutorial video of Niels Biology, and in this video tutorial I am going to discuss about Archaea. In the previous video we have discussed on three domains of life that consist of bacteria, eukarya and archaea, differences of their structural and functional features. But in this video I am making a dedicated video on Archaea, what you need to understand. This is a typical archaeal cell. Archaea cell consists of a cell wall, a lipid monolayer, and their nuclear materials. It does not have a formed nucleus which present in eukaryotic cell. This present in it is nucleoid. Their nuclear material looks like nucleus but does not resemble to nucleus, and they have plasma membrane and cell wall, consist cellular proteins, and last is flagella. So what is the structural composition of their cell wall? Their cell wall normally made up of pseudopeptidoglycan. Pseudopeptidoglycan means this is not a peptidoglycan which is present in bacterial cell. Pseudopeptidoglycan have a different features. They does not have D-amino acid and N-acetyl-muramic acid, which is structural composition of peptidoglycan. This does not present or resembles in archaeal cell. So in case of cell membrane, the cell membrane have branched isoprene hydrocarbon chains. Hydrocarbon is attached with glycerol via, need to use a different marker for your better understanding, ether linkage. Why archaea present in this extremely desiccative condition? Because they have branched hydrocarbon chains in their cell wall, branched hydrocarbon chains in their lipid, which is attached with glycerol by ether linkage. Okay, so these features, structural features, is the main theme by which they can survive in extremely cold or extremely hot condition. So I will discuss about methanogens, halophiles and thermoacidophiles. Methanogen can make methane gas by degrading substance present in marsh area and also gut of ruminant animals. Methanogens, example, Methanopyrus candleri. Methanopyrus candleri. An example of methanogens. Methanogens, these are obligate anaerobic autotrophs. They live in oxygen stressed condition. They cannot live in oxygen, full of oxygen area where oxygen is present. They cannot survive in those area. Methanogens can form methane. If there is acetic acid, they can form acetic acid into methane, or they also used to form gobar gas. This is industrial value of methanogens. NEET and your exam, these questions frequently can come. They used in gobar gas. Halophiles found in extremely salty condition, where salt concentration varies from 2.5 to 5.0 molar concentration. Halophils can live in extremely salty conditions, and halophils have bacteriorhodopsin, bacteriorhodopsin, which acts as their pigment by taking sunlight or ray. It looks like purple in color. Bacteriorhodopsin. It also helps to form ATP. ATPs help in extremely oxygen stress condition. ATP can help halophiles. Halococcus can be a good example of halophils. Question frequently comes in competitive exam. In which category of Archaea bacteria rhodopsin found? The answer will be halophils. Okay. So these are different features. They are actually aerobic, but facultative anaerobic condition. Facultative anaerobic heterotrophs. They found in salt lakes, or their salt concentration is very high. Their ribosome and RNA polymer can work in extremely salty condition. The last one is thermoacidophils. Example of thermoacidophils: thermoplasma. It finds in coal mines in extremely hot condition, extremely hot or extremely cold. They can absorb heat up to 80 degrees centigrade, and in minus it might be 50 to 70 degrees centigrade. Or they also found in volcano where temperature is also high, little bit high from 80 degrees centigrade, but this is the average value. Thermoacidophils, they uses chemical energy to form their food, so they known as aerobic or facultative anaerobic chemoheterotrophs. They are chemoheterotrophs. Oxygen is needed for their survival, so they are heat lover. So these are the difference between methanogens, halophils, and thermoacidophils. Hope you like this video. If you like this video, then please subscribe to my channel. Stay happy and keep learning. We will back in another video to understand how to differentiate, or how to differentiate various types of taxon or taxa for understanding. So till then, stay happy, keep learning. Thank you.